Hi, my name is Jessica Redline, and I'm going to be demonstrating Lab 3 for Micro 225 for LAPU, and today is 7/17/2020. So first I'm going to start off with dissecting my area with 10% bleach. And then I already pre-poured my plates. I have my agar plates ready, and they're all labeled 10-1, 10-2 through up to 6. I have 6 agar plates, and so I pre-did that, and I recorded that in the video before on how to set up the agar plates. Usually I did mine about an hour before, and I let them sit and cool with the lid tilted off like that. And so now it's all molded and ready to go. It doesn't move around. And I had it facing down like this. And then I have my alcohol and my pipette here. I have my yeast, which has been sitting in warm water for about 10 minutes. And I also have my test tubes labeled from 10 to 10-1 to 10-6. I don't know how I can get them to stay properly, but let me stand them up really quick. Okay. It's hard to get them to stand up properly because they're a lot smaller than the actual hole size, but they're all labeled. I already pre-poured them with well, actually I only pre-poured the 0.25, so I'm gonna add 2 mLs to it now on camera. So let me get a little bit of water. So I have my water cup. I have a clean pipette, and I'm just gonna add about I'm just adding 2 mLs. I already added the 0.25 to it, so you want to add a total of 2.25 mLs of distilled water to each. Test tube. So just like that. I'm gonna clean my pipette with alcohol for about 20 seconds. It says to just draw a small amount of alcohol into the tube and then expel the alcohol into the sink. And then it says to just expel any excess to make sure that the pipette is dry and free of any alcohol. I'm just doing that onto a paper towel. So I'm gonna swirl my yeast solution and it says to pipette 0.25 mLs of the yeast solution, also known as Cerevisiae solution, into the test tube 1. So I'm gonna go ahead and do that now. And then I'm gonna make sure all the solution is in there. Now I'm gonna pipette 0.25. It's hard without getting into, so you're gonna draw from 10-1 to 0.25, and I put it into test tube 2. And I'm just gonna make sure all of it is in there, and then I'm gonna repeat the transfer process. So I'm gonna get I need to find the line. So now we're in tube 3, and I'm gonna transfer to tube 4. Now from tube 4, we're gonna go into 5. And then from tube 5, we're gonna go into tube 6. So that completes the transfer process. And then I'm gonna rinse the pipette in distilled water. Just to remove all the organisms out. And now we're gonna be inoculating the plates. So now that I'm just cleaning my pipette, I'm going to get all my plates here. So I'm gonna get plate 1. I'm gonna get approximately 125 mLs, which is 4 drops, and I'm gonna put that into the agar plate 1. Okay. And then I'm gonna put the cover back on and repeat the process. So we're gonna go into pipette 2 with our plate 2. And I'm gonna place the cover on that. I'm gonna rinse my pipette. Make sure that it's dry. We're gonna go into 3. Just like that. Place the cover on. Then we're gonna go into pipette 4. Place the cover. And last but not least, we're on 6. And we're gonna place the tube on that. Sorry, the cover on that. So it says to swirl, so you can move around the yeast on them. So that's what I'm gonna do. Kind of move it around on the agar. And after that, I'm going to dispose of the yeast on the drain, and I'm going to clean the test tubes with my soap and water and return it back to the test kit for further use, and also sterilize the pipette so that I can use it for further labs. And I'm going to place my my inoculated plates. I'm going to place them for, with the lid. It says to put in a location where I'm going to incubate, which is, I've been using the area above my microwave, where I have like a little basket and it's dark. And I'm going to keep it in there for about 12 hours so that the liquid can be absorbed. And then after 12 hours, it says to invert the dishes and incubate for an additional 36. So after a total of 48 hours incubation time, I'm gonna check the plates for any S. cerevisiae colonies and see how well they've developed. And it says that there should be, it should show a growth pattern. So we will see then. So I'm gonna just follow the instructions and let these guys start to incubate. Thank you.